All right, everyone. We're going to go ahead and get started. Thank you so much for being here and being a part of this community. Science is amazing, and I'm glad you're all here. I'll just say it out loud. So my name is Cinnamon Moffat, and I'll be your host for today. This is a hybrid event, so we have folks online, folks here in the room. If you are new to these events, I'll give you just a couple logistics. For folks online, your cameras, mics, and screen shares have been disabled, but we have a volunteer in the room, Roseanne, who will help navigate any questions you might have technically, and will read out any questions you might have for today's speaker. So just put those into the chat box, and then you can communicate with us that way. For folks in the room, when we get to question and answer, because we have folks online, you will need a mic to be able to communicate with them. So raise your hand if you have a question, and I'll bring you the mic, and then you can ask that question for our speaker for today. But again, thank you so much for being here and being a part of this. I wanted to make a couple of quick announcements before we jump in. So we do these seminars every single week. So next week's seminar is February 27th, when we have Gordon Cruz from the University of Alaska Fairbanks here to talk about ecosystem-based fisheries management of crab fisheries in the Bering Sea and the Aleutian Islands. So that's a very timely topic that I'm excited to have him join us and talk a little bit more about what's going on up there. For everybody else, if you are curious about our seminar events, you can go to the HMSC events page on our website, scroll down, and you'll see a calendar event there. You'll get information about what's coming up and how to join us if you'd like to for those. But I want to get going with today. We have a great speaker here. We have Zach. Zach, I didn't ask you how you pronounce your last name.
Meunier.
Meunier is a postdoctoral research associate in the College of Earth, Ocean, and Atmospheric Sciences here at Oregon State University. His current work focuses on applied machine learning methods to predict regime shifts in marine ecosystems, particularly coral reefs. Zach has earned his PhD in integrative biology with a minor in statistics at Oregon State University, where he was co-advised by Bruce Mangy and Sally Hacker. With fellowship support from OSU and the National Science Foundation, Zach studied rocky intertidal ecosystems in Oregon, California, and Nova Scotia. His interest is in how climate change, disturbance events, diseases, and species interactions influence community structure and dynamics. And I am very excited to hand the mic off to Zach. Go ahead, floor is yours.
All right, thank you, Cinnamon, for the introduction. And thank you, everybody, for joining us today for this seminar. So I'll be describing some of the research that I conducted as a PhD student at OSU, and then I've continued to conduct as a postdoc at OSU. And the title of my talk is Regime Shifts or Resilience, Causes of Change and Consequences for Diversity on Rocky Shores. So with that, seems to be frozen. There we go. All right, with that, I have two major themes for my research. The first is to assess the causes and consequences of changing coastal ecosystems. And I work primarily on two ecosystems, rocky intertidal habitats and coral reefs. And I've also done field work in lakeshore dunes in Wisconsin as an undergrad, and I've done some modeling work on kelp forests. And then the second theme of my research is to develop and apply novel statistical and machine learning methods to detect and predict changes. And I do this primarily in two programming languages, Julia and R. And for my postdoc work, I'm doing what's called scientific machine learning, where we have a small amount of training data and then some model, and we can extrapolate from those training data very far and accurately into the future. I don't have too much time to talk about the scientific machine learning today, but that is also part of what I do. So these are the research themes. And then I also do research that addresses climate change, getting into the motivations for why I do what I do. So climate change, as we all know, is a very existential threat in many ways. For the oceans, it's particularly threatening in terms of the temperature, pH conditions, and oxygen conditions for the ocean. In my own research, I focus primarily on the warming events, and especially on the abrupt warming events. But of course, with global warming and the chronic warming in general, lots of ecosystems are under threat. So this is the first research motivation. The second is this idea of shifting baselines, which is how a system is measured changing over time compared to its reference point. And that's because that reference might be ignored intentionally, forgotten unintentionally, or unknown to begin with. And to give you an idea of what this might look like, I have here a time series of the number of hot days per year. These data are taken from Germany, so it's a temperate area, 86 degrees Fahrenheit, 30 Celsius. Above that is hot in this area. Now, folks born in generations previously, like baby boomers, Gen X, they experienced on average 4 hot days per year. I'm a millennial, and so folks in my generation would have experienced 6 per year. And Gen Z generally is experiencing 9 days per year. So this idea of shifting baselines is that on average, it's increased over time. And if you are only thinking about what you yourself have experienced, you might not have that historical context. And this results in what's known as shifting baseline syndrome, where the inability of people to perceive changes because of lost knowledge about past conditions. And it's also known as generational amnesia. And this happens also in the oceans. So for example, in the 1800s, a diversity and abundance of marine life was very common in coastal oceans. The 1950s declines in the diversity and abundance. 2019, for example, lower diversity, lower abundance, and plastic pollution. And if you're comparing, say, 2019 to 1950, the change might not be as dramatic as if your baseline was the 1800s. And so with that, I want to address one more core concept before I get into the details of my research, and that is this idea of resilience and regime shifts. So we can imagine some environmental condition that changes over time. And for example, in the first period of time here, we have some lower mean condition. In the second period of time, it increases into a higher mean. In the third period, back to that original mean condition. And so I've simulated this time series just as an example, but it's on average the same in periods 1 and 3. And these conditions, it might be temperature, affect what we observe in the ecosystems, the communities. So for example, in the first period of time, you might have a base community state that is a grassland. And that grassland might resist the temperature increase and maintain itself as a grassland ecosystem. And then with the return of these colder temperatures, it may still remain a grassland. We call this persistence over time resistance. And this is in contrast to a system like a grassland switching to a desert with warming conditions, and then switching back to a grassland with cooling conditions. And we call this recovery, where we go back to that original alternative state. And these two are collectively called resilience. And resilience is in contrast to a third type, where we have a base community state or alternative state, such as a grassland, that transitions to another community, like a desert. And then even though the environmental conditions have reversed, that community is maintained in this second state. And we call this a regime shift, where even though the underlying environmental conditions have changed, the community itself has not changed. So this is a little bit of a conceptual example, and then to apply it specifically, I work in rocky shore habitats, which are characterized by a variety of temperature and other abiotic conditions that vary over a gradient from low intertidal, that is closer to the ocean, to high intertidal, a little bit higher elevation. So for example, in the low intertidal zone where I conduct most of my fieldwork, the temperature variation is lower, there's less salinity because it's mostly less salinity variation because it's mostly covered by the water, and the competition is generally higher than in the high intertidal because of the number of species. So we also have gradients in intertidal species diversity. The lower on shore is generally the higher diversity. And because of the presence of predators being more abundant, we generally have higher predation pressure on the lower shores. All right, given all this information, I'm going to present today 2 case studies. The first is some of my PhD work to measure changes in community types and relate those changes to environmental factors. And the second is to describe the consequences of change for the invertebrate and algae diversity on rocky shores. So the first case study, causes of change on rocky shores. I do my fieldwork up and down the Oregon coast and also in Northern California. This is a photograph from our field site, Rocky Point, which is near Port Orford. In the background is Redfish Rocks Marine Reserve. So this is a place that's near and dear to my heart. And this study is published in Nature Ecology and Evolution. It was published in June of last year, and this was part of my dissertation work. I published this with my PhD advisors, Bruce Mengi and Sally Hacker. Now, this study takes place at 13 field sites, 10 in Oregon and 3 in Northern California. So for reference, today we are here in Newport, and if you were to drive a little bit north to Depot Bay, you'd be on Cape Foulweather, where we have 3 field sites. A little bit farther south to Yachats and Florence area, that's Cape Perpetua. We have 3 field sites there. We also have 4 field sites in Southern Oregon on Cape Blanco and 3 in Northern California near Cape Mendocino. So altogether, 650 kilometers of coastline. And for today's talk, I'll mostly be referencing the capes because they are representing a cluster of sites. So we have lots of spatial replication due to the cape nature. I also want to acknowledge that I have the privilege to conduct this research on the homelands of many different tribes. So the context for today's results are really this idea of warming oceans globally. And I talked a little bit about chronic warming earlier, but my research focuses primarily on abrupt warming, such as that associated with marine heat waves, which generally is when warm water temperature can persist for weeks, sometimes even years, though, as was the case in the blob, which was a very large event on our coast. And I'm using this definition that's become a standard where the temperature exceeds some 90% threshold above the average sea surface temperature. And so I mentioned the blob, which was a massive marine heat wave that really dominated the Northeast Pacific Ocean and all the way up into the Gulf of Alaska. And it lasted on and off from January 2014 through August 2016. And it resulted in the mass mortality of many different species, including the seabirds pictured here and the kelp that I study in the intertidal. And this is a series of data from the rocky intertidal. So these are sea surface temperature anomalies on the y-axis. The positive anomalies are warmer than average, that is above the 0 line. Negative anomalies colder than average, below the 0 line. And we have the study period from 2006 through 2020. And I have our 4 study regions as the 4 colors, which I'll be using throughout this presentation. Now, the marine heat wave, the blob, was really, really strong warming from 2014 through 2016. And this was unprecedented in the history of this time series. So we have actually 30 years of data to base our long-term mean on. And the marine heat wave was excessively warm for several years. So this is one of 2 major stressors in the system. The other is sea star wasting disease, which I'm sure many of you are familiar with, afflicts many different species of sea stars, in fact, more than 20. On Oregon coasts, the most important sea star afflicted is Pisaster ochraceus. And the symptoms here progress rapidly from tissue loss and decay to body fragmentation and eventually death. Here's a picture of the Pisaster, the ochraceus star, the keystone predator, on the first day of its disease. And there's a lesion on the upper surface here in the center. And then also the arms are twisting and unable to connect to the rock. By the second day, the arm has fragmented from the body and the sea star is no longer attached. By day 3, the body is completely disarticulated and no longer able to attach at all. And it's very likely the star is dead. So this resulted in, unfortunately, a mass mortality event of these ochraceus stars that began in 2013 and 2014 and persists today. And there is some evidence that the disease outbreak was exacerbated by the marine heat wave, but I want to stress that the marine heat wave did not cause the disease. The disease was actually occurring before the blob on the Oregon coast. So this is a time series of the prevalence of the disease in Pisaster populations from our long-term monitoring. So we have on the y-axis the percent of sea stars afflicted, and on the x, the same 15 years that I'll be showing throughout the presentation. And the vertical dashed line is the outbreak of sea star wasting disease. So before that outbreak, we did not detect the disease presence. Afterward, over 80% of the sea stars in some of our study locations had sea star wasting disease. Because of the disease's virulence, it kills most of the adult stars immediately. And then we see a crash in the number of sea stars that are afflicted. But you can see from 2015 through 2020, that the disease prevalence is still somewhere between 15 and 20%. And I took this photograph in 2022, so 2 years after these data that are presented. You can still see several disease stars here, some lesions on their upper surface, the arms are twisted. And this, again, is the major predator in the system. So it does have a very important influence on the system. Now, the disease resulted in declines of the abundance of Pisaster per unit area, which we call density. And after the disease outbreak, we saw an immediate decline in density, but you can actually see some recovery after that period. So especially in Cape Perpetua in Central Oregon, there were a lot of sea stars present, but these were mostly baby stars that had just recruited. All right, given all this environmental context, I want to introduce you to my favorite experiment, which we affectionately call CRAZY. So not only is it crazy because it spans 650 kilometers of coast, but also because it's a fun acronym for the Community Recovery in the Algal Zone Interaction Experiment. And CRAZY consists of 5 blocks that are replicated at each of our 13 study sites. Each block has 4 treatments. And those 4 treatments are a control treatment, as pictured in panel C, where we never manipulated it, a recovery treatment that was initially cleared in 2006, and then we allow species to come back naturally, and then 2 annual removals, initially cleared in 2006, annual removals of invertebrates from the macrophyte-only treatment, or macrophytes from the invertebrate-only treatment. And a macrophyte is the surfgrass and the kelp and other algae. So anything that is a primary producer is a macrophyte in this case. So altogether, we have 260 fixed plots, and we've been tracking these for 19 years and counting since 2006. So we, being my advisors and I took this project over in 2017. I was in middle school in 2006, so I wasn't able to do it then, but I took these 12 years of data and I've run with this project ever since. So given this, I can pose 4 research questions. The first is, what were the changes in rocky intertidal community structure as a result of our stressors, the disease and the marine heat wave? The second question is, where did regime shifts occur? And were they associated with our 2 stressors? Third, what mechanisms were responsible for maintaining the alternative states? And fourth, were any of the communities resilient to these stressors? So lots of questions here. In order to answer these questions, we conduct community surveys. And I want to give you a sense of what that looks like. So we go out in the field, and this is one of our experimental plots in the control treatment. So again, never manipulated. We find the bolts that mark the corners of the plots because these are fixed plots over time. And then we determine the abundance of the species present. So for example, we have mussel cover at about 15%, kelp cover about 25%, a few species of red algae, Cryptopleura at 8%, Microcladia at 10%. And we do this for every species present. And we have now repeated this 4,722 times. And I myself have done nearly 2,000 of these surveys. So it's a lot of work, but it's a lot of fun, and it gets me outside. So before I get into the data themselves, I want to give you a sense of what the changes looks like in photographs. So we have in Cape Perpetua, the central coast, Cape Perpetua is normally dominated by invertebrates. And so we get a lot of invertebrate recruitment here. This is a recovery plot that was initially cleared in 2006, 3 years later covered in acorn barnacles. 5 years after that, still covered in acorn barnacles. And again, this is right before the disease outbreak, right before the marine heat wave. Now, after those events, it's dominated by gooseneck barnacles. And gooseneck barnacles are like the popcorn for sea stars. They love to snack on goosenecks. So this is some evidence, at least anecdotally, that the sea star absence might have allowed these goosenecks to recover and really dominate this space. 3 years later, you can see there's a Pisaster foraging on the goosenecks in this plot. So this is the increase of gooseneck barnacles in photos, but let's look at the data here. So we have a time series of gooseneck barnacle abundance averaged in the treatments where it can occur at our 4 study regions. And you can see that in Cape Perpetua, Central Oregon, a dramatic increase in gooseneck barnacles by a factor of 7.4 times immediately after the disease outbreak and the marine heat wave, but it has declined a bit since then. All right, a few more photographs now. We're zooming to our southernmost field site, Kibitz Hill in Northern California. This area is generally dominated by kelp. This is another recovery treatment plot initially cleared in 2006. 3 years later, dominated by kelp. 5 years after that, still dominated by kelp. And then the marine heat wave hits, sea star wasting disease happens. We get these transitions from kelp-dominated to mussel-dominated habitats. And now the mussels are so dense in this area that I have to scramble around with a metal detector to actually find the bolts. So I'm going out in the middle of the night on my hands and knees using a metal detector, poking around. It's a good time. All right, so let's look at some data. Here we have the kelp abundance over time in our 4 study regions. And we can see that in Southern Oregon in pink and in Northern California in orange, there were dramatic declines of kelp. Again, during the marine heat wave event, kelp declined in Northern California by a factor of 3.5 times. This is a time series of mussels. And so mussels generally increased everywhere, but especially in Cape Mendocino in California, where they increased from near zero to over 45% cover, which was a factor of 826 times. So given all this information, we can answer this first research question. What were the changes in rock intertidal community structure? In some areas that were previously dominated by algae, we saw transitions to dominance by the sessile invertebrates. And in general, we saw some of the favored food, the mussels and the gooseneck barnacles, come back in places where they were not previously dominant. So moving on now to the second question, were any of these changes actually regime shifts? And were they associated with our 2 stressors? So let's investigate the causality behind this. In order to answer this question, I did a suite of multivariate methods. And so I want to go over those at a high level. The first is called cluster analysis, which essentially is taking every single plot at the tips of a very large dendrogram. So near the bottom here is many, many, many experimental plots. And then we organize these plots based on how similar they are to each other, kind of like an evolutionary tree. And then we cut this dendrogram in certain places to give us different groups and different numbers of groups in varying sizes. So that's cluster analysis. The second is indicator species analysis. And this actually allows us to figure out which species or functional groups are important for distinguishing our different clusters. So for example, this plot here is dominated by mussels, and the mussels would be very important in the indicator species analysis results. So collectively, we use these two to go from a very large matrix of over 100 species to a little bit smaller matrix of about 10 what I call community states. And these are dominated in general by a handful of functional groups. And I've color-coded the functional groups as follows. So the colder colors are for the invertebrates. The warmer colors are for the algae. And then, of course, grass is green. Surf grass is in green. So this photograph here is a control treatment plot, and it's actually this little tile in this diagram. So we have time on the X, our 15 experimental plots in Cape foul weather for the control treatment on the Y, and then they're color-coded by their dominant functional group over time. So this is one treatment in one Cape, but I have 4 treatments and 4 Capes. So I can do this for all of them and make a very fun diagram that there's a lot to unpack here, but I'll go through it slowly. So again, on the X-axis, we have time. On the Y-axis, we have the relative abundance of our different community states, and there's 10 total community states, which are given at the top as these different colors. And then they're organized across the columns by treatment and down the rows by the Cape from north to south. So in the northernmost Cape, we saw increasing mussels in dark blue in the 2 control and recovery treatments, generally after the disease outbreak in both of these cases. In Cape Perpetua, Central Oregon, we saw increasing gooseneck barnacles, especially after the disease outbreak in the control, recovery, and invertebrate-only treatments. Declining kelp in Southern Oregon. So we have lots of kelp pre-marine heat wave, much less kelp afterwards in our 3 treatments where kelp can occur. And then finally, in Northern California, we have this transition from kelp abundance here, then the kelp decline, to mussel abundance after these stressors occur in the system. All right, so that's one part of it. The second analysis is a multi-level hidden Markov model, which is a complicated multi-dimensional analysis. But generally what I'm trying to do is go from a lot of information in a very high dimensionality to a much more simple understanding of it. So I do that via a transition probability matrix in the middle. So I'm going from what we can observe in the community to what might be hidden, that is the alternative states. So we start with a time series of our different community states in our 4 treatments. The multi-level nature refers to the fact that I'm conditioning on all 4 treatments at the same time. So we have our control, recovery, macrophyte only, and invertebrate only treatments. And you can see that they're generally having some change. You can see some warm colors on the left, cool colors on the right. But is this actually what we call a regime shift? So we do an intermediate step where we have a transition from alternative states 1 to 2, and what are their likelihoods. So this shows the probabilities of going, say, from alternative state 1 to alternative state 1 are fairly high. That is, this bar is really thick. But to go from alternative state 1 to alternative state 2, it's much less likely. So this is more of a thin line here. And this gives us some one-dimensional output. So we go from a 4-dimensional input to a 1-dimensional output. And we can classify things into one of 2 alternative states. So for example, this particular replicate went from alternative state 1 in the first 10 time points to alternative state 2 in the last 5 time points. And this is a Bayesian analysis. So if you're not familiar with Bayesian statistics, it's a different way of doing statistics than what we think of as the frequentist p-value based stats. Generally, we have some evidence, that is our data, and some prior information about the system. And we average those 2 using Bayes' theorem to go from prior beliefs and evidence to some posterior distribution in the middle. So the blue curve here is a weighted function of our red and black curves. And so what I'm going to show you is some posterior distributions where we have the probability of observing community state J in alternative state I, given that we're in one of those 2 alternative states. All right, so I'm going to state that we have regime shifts in 2 out of 4 regions, and I'm only going to show those diagrams, not from the regions where we didn't observe regime shifts. So we saw regime shifts in Cape Perpetua, and I'm defining that based on a transition in the abundance of what our alternative states are. So that is where the lines are crossing here, here, and here. We have a transition from one alternative state being more abundant to the other being more abundant. And I've done this for the 3 sites in that area. And I've categorized these 2 alternative states based on the functional groups that really represent them. And so in order to look at which functional groups are important, we take a look at the posterior probability distributions. On the left panel, this is alternative state 1. This is dominated mostly by rock, and then all these other functional groups are less abundant. They have less probability of occurring. The right panel, alternative state 2, we have this gooseneck barnacle distribution that is much farther to the right than all others. And this is the control treatment. We see the same thing in the recovery treatment. We go from a rock-dominated state 1 to a gooseneck barnacle-dominated state 2. So this is evidence of regime shifts. In 2 out of 3 sites, these regime shifts happened after the disease outbreak and marine heatwave event. All right, now on to Cape Mendocino in Northern California. We see here the same kind of image, but in this case, all of our transitions are happening after the disease outbreak and marine heatwave event. Alternative state 1 in the control treatment is characterized by kelp and branching and bladed algae, and alternative state 2 by mussels. And then the recovery treatment, we see the same thing, transitions from kelp and branching and bladed algae to mussels. All right, so this allows us to answer the second research question. We did see regime shifts, and in 2 out of 4 capes, they were associated with our 2 stressors. Moving on now to question 3, what were the mechanisms that were actually responsible for maintaining these alternative states? So I went out to do more field work. I thought I was done in 2020, but turns out I had more hypotheses about what was going on. So I went out in the field at my advisor Bruce's suggestion to measure the mussels and the sea stars that were occurring in our plots. So this is an example of me measuring one of the mussels that was particularly difficult to get to because its posterior end was under a bunch of gooseneck barnacles. And the reason I was measuring these mussels and the sea stars is because there was a very strong response in the decline of the size of sea stars across all 4 of our study regions. So right after the disease outbreak, you can see here, every region, the average sea star size crashes dramatically. And it's recovered somewhat in Northern California, but has not yet recovered as of 2020 in these other 3 capes in Oregon. And I was measuring mussels and sea stars because my advisor's advisor, Bob Payne, published this really cool paper in 1976, where he defined the relationship between the average mussel and the average sea star size. So this is, every point here is an event where a sea star was feeding on a mussel. And you can see a high density of points in this area. That is smaller size stars feeding on smaller size mussels. There's a lower density of points above, and that is there aren't as many big stars, and generally they're going to go for the really big mussels to make it worth their while. And Bob Payne defined an upper limit of predation that is the solid line, above which the mussels are too large to be eaten by Pisaster of a given size. So he defined this relationship in 1976 based on many, many feeding events. And I went out in the field and measured the size of the mussels in our plots. And in 36 out of 39 cases, I found that the mussels had grown large enough to escape predation from sea stars at a given site. So this is, what is the average sea star size at a given site? In all cases, the mussels are so large, except for 3 points that are just on that line, almost above the upper threshold. All right, and this is really important because mussels can live for up to 30 years. That is, if they grow large enough to escape predation, they can be there for a long time, and that makes these alternative states very persistent. That was the impact of sea star wasting disease. The other major stressor is the marine heat wave, and we're interested in what was the impact of that on kelp. This is a very hairy table that I don't expect you to read, but it's important because it shows the thermal tolerances of many different algae species in the Pacific Northwest. Near the top of the table are all the algae that are cold tolerant. The bottom are all the warm tolerant species. And it turns out that all the intertidal kelp are near the top, especially Hedophyllum sessile, the most abundant. That has an upper threshold of 15 degrees Celsius. And so then I regressed the kelp canopy cover on the y-axis onto the water temperature anomalies on the x-axis. On the far right here are the marine heat wave anomalies. That is, these anomalies were only observed during the marine heat wave event, and we see strong negative declines in Cape Mendocino and Cape Blanco of kelp with these temperatures. All right, so that answers the third question. I would say that the mechanisms that are really important are the lack of keystone predation for several years and the extreme temperature stress that these organisms experience. Finally, were any of the communities actually resilient to this change? So one more analysis for this section here. This shows on the x-axis our 4 treatments, and within those, the 4 different study regions. On the y-axis, the community similarity. So near the top, we have communities that are really similar over time. Near the bottom, communities that are really dissimilar over time. And in this case, I have compared the plots to themselves for all possible combinations of time. So a plot is only being compared to itself, not to any others, which is important for consistency. And generally, the only ones that had pretty high resilience or resistance are near the top. In this case, this is the macrophyte-only treatment in Cape Perpetua. And in general, macrophytes don't occur in Cape Perpetua in our experimental plots. So in this case, it's actually just a signal of rock, maintaining itself as rock over time. So it's not actually resilient there. And then I have 3 colors. Blue is the pre-disturbance conditions. Red is the resistance. And orange is the resilience. So the blue is what was happening before any of these stressors. If the red and the orange are different from blue, then we have significant differences such that we have either a lack of resistance or a lack of resilience. So in 10 out of 16 cases, we had low resilience. That is, we had significant differences between blue and red. And in 10 out of 16 cases, we had low resilience, significant differences between blue and orange. Cape Mendocino, where we saw regime shifts, was the one that was affected the most. So there are the biggest differences between the blue dots, the red, and the orange dots in our 3 treatments, control, recovery, and invertebrate only, where the mussels can occur. All right, so to wrap up the first case study, we have regime shifts occurring as a result of temperature stress on macrophytes, reducing their populations. At the same time, we also have a disease outbreak for the Pisaster ochraceus, the keystone predator, resulting in a population decline for them across the board. This allows their prey, gooseneck barnacles, mussels, acorn barnacles, to increase in abundance. And that gives us regime shifts in some of our study sites from macrophyte-dominated to invertebrate-dominated systems. All right, so I talked a lot about regime shifts at a very high level. Now, what were the actual consequences of those regime shifts for long-term species diversity? So the study, the first one that I presented, was through 2020, but I've continued, as I said, to monitor this project for 4 additional years. So this is one of our study sites in Northern California, the one that observed a lot of regime shifts. And you can see the mussels are really starting to creep down from the middle intertidal into the low intertidal zone. Now, there are many ways of assessing species diversity. The 2 primary metrics that people measure, though, are species abundance, which is how many individuals of a given species are present, and the richness of species, that is how many are present in a given area. And it's important to measure these things because there's a very clear relationship that is honestly a textbook example that I had in my biology textbook. I'm sure you all are familiar with it. In the absence of this keystone predation by Pisaster, Bob Payne showed that there was a decline, the red line, in the number of species present from 1963 to 1966, and then that was maintained for a number of years afterward. Conversely, with Pisaster being present, there was a maintenance long-term of the abundance of a variety of different species. So the same number of species. Here, this does not take into account their actual abundance, though. This is just the richness, how many species were present. And so in my research, I want to go a step further and actually look at the trends in abundance. For time constraints today, though, I'm only going to have time to talk about the richness, but future work will address the abundance question as well. And there's many ways of measuring species diversity also with regard to their spatial location. So there's what's known as alpha diversity, where you have distinct sampling units, in my case, the different plots. And then there's beta diversity, which is the difference among those plots, the turnover in species. And then if we zoom out a little bit, we get what's called gamma diversity, which occurs at a more regional perspective. So we might think about the sites or the capes themselves as the gamma diversity area. And I'm measuring diversity using this formula, which is the Hill number framework. This D stands for diversity. Q is the index, which adjusts the exponents here. And very simply, the index shows when Q is 0, species richness, when Q is 1, Shannon diversity, and when Q is 2, Simpson diversity. So again, species richness is only looking at the identity of the species, not their abundance. These other 2 indices look at the abundance, and then they weight the common species more as Q gets higher. But I'm only looking at the first one, species richness, today. So I'm going to show you a series of plots. The first slide here will show richness per plot. So how many species are occurring per plot? And I have means and 95% confidence intervals. So our 4 study regions, within those are 4 treatments. In all cases, we see declines from 2014 through 2017 in species richness. So during our stressors, we saw declines in the number of species present. This is not their abundance. And we also saw after that, from 2017 through 2024, there seems to be a rebound, right? So all the curves are then increasing after that period of time. And this is across all 4 treatments. So it seems to be something quite systemic. So I wanted to dig into that a little bit deeper. Which species exactly declined, and did the same species come back? And so I broke the time series into 3 periods. I'm ignoring the first 2 years because of our experimental clearances in 2006, and I wanted to make sure that some of the successional processes were done. That is, a lot of the early arriving species had arrived already. So I'm ignoring 2006 and 2007. The first period is the 7 years from 2008 through 2014. The next period is 3 years during this lull in richness. And then period 3 is from 2018 through 2024, another 7-year period. And I'm only going to focus on Cape Mendocino again for time. All right, so I'm going to show a few more plots here. On the x-axis, I'm going to show the difference in functional group occurrence between sampling intervals per plot. What does that mean? So for species richness, again, we're not concerned with abundance. We're just concerned with presence absence. That is their occurrence. And we're looking at the mean occurrence per plot. So over time, within these sampling intervals, how often did a species occur in a given plot, averaging all the 1s and 0s. And then we're taking the difference between sampling intervals. So periods 1, 2, and 3, what are the pairwise contrasts among those? First, mobile invertebrates in our 4 study treatments. I've color-coded red for declines, significant declines, blue for significant increases, and gray is no significant differences, no significant changes. In this case, we have 3 functional groups that declined and none increased, and this is going from before our stressors to during, so periods 1 to 2. We see declines in 3 of our functional groups. We can also now look at the sessile organisms. So at the top 7 are the invertebrates, the middle 2 are the crustose algae, and the bottom 7 are the macrophytes, the surf grass and the different algae. And you can see that in general, there's a lot of red, right? So we had a lot of species that declined in their occurrence. The only ones that increased were the invertebrates, the anemones, the mussels, the gooseneck barnacles. So this is from periods 1 to 2. And now let's look at the rebound from periods 2 to 3. So this is actually showing that a lot of species did come back. In fact, more mobile invertebrates increased their occurrence than actually declined initially. And so we had increases in urchins, sea stars, worms, isopods and amphipods, limpets and sea slugs, and chitons in some, but not all treatments. And we saw a lot of increases, in this case, in our sessile organisms, especially the invertebrates in the algal crusts. So this was from periods 2 to 3. Now let's compare the before and after. Let's look at the net effects of our change. So one more set of plots here. This is from 2018 to 2014, and then comparing a decade later. We see actually mobile invertebrates largely increased in their occurrence by a little bit. The only one that declined was urchins in the control treatment. And for the sessile organisms, we see blue on the top, red on the bottom, indicating that we gained sessile invertebrates and lost some of our macrophytes, especially canopy-forming kelp and the understory articulated coralline algae. All right, so I can put all this together in a little bit of a scorecard. And this is the simplest math that I'll share today, just straight up addition. So I've color-coded them. Did we gain or did we lose functional groups in these different periods among these different areas? So in sessile invertebrates case, for every different sampling interval comparison, we did gain functional groups. The net effects in the last column show that we gained in the occurrence of functional groups by 3. For mobile invertebrates, we got this initial decline in the first period, first change, and then they rebounded, giving us a net effect of a gain of 4 more occurring functional groups. Crustose algae declined initially, then rebounded, and that was net neutral for the number of species present, and macroalgae declined in all 3 of these comparisons. So ultimately, we had some winners and losers. We had the species numbers rebound, but it was not the same species. We had declines in kelp and urchins and articulated coralline algae and other macrophytes. And we had increases in invertebrates like mussels, gooseneck barnacles, acorn barnacles, and limpets. All right, so putting it all together, Today in my talk, I described a wealth of ecological data, and I tried to glean insight from it using ecological data science as a framework. And in part one, I discussed the abrupt changes that we observed due to regime shifts in 2 out of 4 capes, going from macrophyte-dominated like kelp habitats to invertebrate-dominated like mussel habitats. And this is actually a photograph of one of my experimental replicates. This little patch used to be all kelp, and now you can see the mussels have completely taken over half of it. That was part one. Part two was focusing more on the consequences. So what, after these changes, did species diversity actually change? We found that the total number of species has rebounded, and at least in Cape Mendocino, the identity of those species has changed from more algae to more invertebrates. So altogether, I think we've learned several lessons through this research. The first is that defining and measuring change is a nuanced science, and that's why I'm devoting a lot of my career to that. Second, long-term studies like crazy are vital for establishing baselines. So I've been doing this project now for 7 years, and I have hopes of continuing it for several decades. Now, large-scale studies like crazy can also show generality and context dependence in their results. So the marine heat wave and the sea star wasting disease outbreak, these were very general events that had widespread consequences, but their impacts were not universally felt in all of our study areas. In fact, we only had regime shifts in the 2 capes that I've discussed mostly today. Capes Foulweather and Cape Blanco did not have those regime shifts. And fixed plots are really important because they allow us to go back and sample the same place year after year, and that ensures that we have a consistent reference point. So altogether, leveraging advanced statistics and machine learning techniques like the hidden Markov model allows us to gain valuable insight from these long-term data. And data science as a framework can help us to understand climate change impacts and prevent the shifting baseline syndrome, but we have to do that work. So with that, I'd like to acknowledge my PhD advisors, Sally Hacker and Bruce Mengi, and funding and support from the National Science Foundation, Oregon State, the Oregon Ocean Science Trust, and the Phycological Society of America. In addition, I want to acknowledge that I had the help of many, many people, including some undergraduate researchers. My brother, who's on Zoom, came out for a couple of summers to help me. My fiance has helped me. So science is really a team effort, and I appreciate the help of so many over the years. And with that, I would love to take your questions.
All right. I'm just going to admit that when I grow up, I want to be Zach. All right. So we're going to switch and have some questions here. We're going to start with questions online, and we have one I can see. So go ahead, Roseanne.
Was tidal strata a variable in your study sites?
That's a great question. So did the elevation vary among study sites? We do have, in all cases, a low zone community that we measured. And we actually have some data on the elevations of the plots as well, because I went out with a surveying laser level. So that allows me to generally get the tide heights of all the plots. And in general, all the elevations across these 13 study sites are within ½ a meter from the lowest point to the highest point, which is fairly good for this level of spatial replication. So pretty consistent tide heights. The tidal amplitude varies a little bit across the coast as well.
Nice. Okay, questions in the room? Yeah.
Great presentation.
Thanks.
One question about, you did say that you saw things were different at the different locations. And you didn't really speak to the reasons for that. And I'm just wondering if you have looked into that, and if you have some thoughts on that you would like to share. Thank you.
Yeah, of course. So we did see regime shifts in the 2 places where I think the organisms were most susceptible. That is in places where the kelp were dominant previously, and they were really sensitive to the stressful conditions. But we also saw changes where the predators were previously really abundant in Cape Perpetua, and their absence allowed their prey to come back. There's a high amount of invertebrate recruitment in the Cape Perpetua area. And so we could have this really fast recruitment event of acorn barnacles, gooseneck barnacles, mussels, and then their growth allowed them to establish long-term. So those two areas were primed in a way for these regime shifts based on the species that were already present. Now, the other two capes, Cape Foulweather is in the plots mostly dominated by surfgrass. So that was a lot of green that I showed for those slides. Surfgrass is actually pretty resistant to these marine heat wave effects, at least so far in our study system. And therefore, it didn't decline, and in some cases actually increased after the marine heat wave. Now, mussels did increase there. And at first, I thought it might be a regime shift. And I have a whole third chapter where I transplanted mussels into and outside of surfgrass patches to see what their interaction would be. And generally, we found that the mussels could not establish in these later years. So we think that it might have been actually the expansion of mussel beds, not just mussel recruitment, that affected them. And then Cape Blanco is just a very variable area. So we have 4 sites there. One of them is invertebrate dominated. The other 3 are dominated by algae, is the only place where all 10 of our community states occurred. And I think there was, in some ways, maybe so much noise that the algorithm couldn't pick up or detect these major changes. I have tracked the kelp populations there, and they have come back. So interesting that they were able to come back in Cape Blanco, but not in Cape Mendocino. And I'm working on another paper that explores that. Didn't get to talk about that today, but I will hopefully have another paper prepared soon.
All right, other questions?
Great presentation, thank you. In the big picture, environmentally, what does it mean for us, everybody, that let's say these areas now have more mussels instead of kelp? What's the impact, or what does it mean?
Yeah, at first I thought it might be a bad thing to have the mussels instead of kelp, because that is the classic example, right, of lost species diversity when you remove Pisaster and then you have mussels present. But in fact, mussels are a very important foundation species, and they have a wealth of invertebrates that live inside of that mussel bed habitat. So from a diversity perspective, you might actually get overall increases in species diversity in these areas if mussels are more dominant. Now, from a human's perspective, I mean, I think it's really just relative to what your preference is when you go out tide pooling or something. Like, I don't think that there is definitely intertidal foraging, but that affects mussels, kelp, a lot of different species in the intertidal. So it's not the most resource extractive habitat. Now, on a broader scale, I think these climate change impacts are only going to accelerate in the next 20, 30 years. And that's part of the reason why I want to continue this study to measure. We have 20 years now of baseline data, some of which has already exhibited a major change. Will it continue to change? Or will these new states, the mussels, for example, be resistant to those changes?
Other questions in the room?
Zach, you got a really impressive data set there. I mean, very long in the context of an ecological data set. And it sort of illustrates how hard it is to pick out these changes that might be recruitment dependent. So I'm wondering how you think about evaluating these ecological periods, like the marine heat wave. You have a few years of where conditions changed, but that event can set up a recruitment event that could have lasting effects that would last for, who knows, decades, potentially for some species after that event has passed. When we talk about regime shifts, do you think that over the long term, it's still going to wind up being a regime shift as the effect of those transient recruitment events kind of work themselves out? Or is that really, that's the scale that we need to talk about, is an environmental event allowing a recruitment event or precipitating a recruitment event?
Yeah, I think that the recruitment piece is another major component of this. My other dissertation chapter looked at recruitment as well. And so I collected plates and toughies on the Oregon coast, which collect respectively barnacle recruitment and mussel recruitment. And I did a study comparing Oregon and Nova Scotia recruitment to see what mechanisms drive that. In general, upwelling is one of the major mechanisms, and also the wave action and the surf zone hydrodynamic conditions affecting recruitment. I think, for example, Cape Perpetua is primed for many reasons to have this really large recruitment. And when I go out and I annually clear what should be a macrophyte-only treatment, and every year it's covered in gooseneck barnacles, I can observe, oh yeah, there's a lot of recruitment happening every single year. Now, my job has gotten a little bit easier the last couple of years because the sea stars are starting to come back, and so they're eating more of those goosenecks, and I don't have to spend an entire day of tide just scraping barnacles out of very small 25 by 25 centimeter section. So that's one aspect. I do think recruitment is important, and it is somewhat variable, somewhat stochastic, but I also see that in places where recruitment is high, like Cape Perpetua, it's everywhere. It's not as patchy. And then I think regime shifts are best measured in reference to the lifespan of the organisms that they affect, not necessarily human timescales. So, for example, in my canned example in the beginning, deserts and grasslands, a lot of those plants that live in these areas can be pretty long-lived, especially deserts, like storing their resources for many, many years. In the intertidal, some of these organisms are very short-lived. And so it is many generations for them if this condition persists for 2-3 years. And in this case, the kelp have been very low in Cape Mendocino now for a decade. The mussels have persisted for a decade. I think that is long enough persistence to call it a regime shift. If it was only a year or two, it might not be as much of their individual lifespans to claim such a regime shift occurred.
Okay, go ahead, Roseanne.
How long was your standard field sampling season?
Yeah, I can get this done if I work extremely efficiently and I have no issues with waves. I can get everything done in one sampling day per site, 13 field sites, 13 days. Tack on a few travel days, it takes about a couple weeks. But during graduate school, the most I did for field work in a summer was about 90 days. Actually, during COVID, because that was my third year of graduate school, and that's when I had to install all my experiments and monitor them. So, it varies, and we can sample on the low tide days on the coast. So we choose the best spring tides to do our sampling. For us, it's in June and July. So those are the months that I really target for my sampling.
Okay, I'm going to ask a question. So can you talk a little bit about the single year class of those large mussels and the long-term impact of that singularity?
Mm-hmm. That's a good question. Yeah, I think what I'd like to do is now go back and measure not only the size of what I did there was the 10 largest mussels. So I chose specifically the big ones, but I also chose them only in the mussel-dominated plots. So I pre-chose based on what I observed in the field. Is this plot dominated by mussels? Yes. Okay, let's find 10 representative ones. Is that still the case? Are they still mussel dominated because of the big ones? Or mussel beds in some ways are positively reinforcing, right? A big mussel will allow small mussels to recruit to them. And is it making more of a mussel bed? So another thing that I could do, and I've got some historical data on this, is the bed depth. So in 2021, my brother came out and helped me. He measured the depths of the mussel beds in the plots where mussels were dominated. And now let's see if they got any bigger, 5 years later. So that's an aspect, because mussels like to attach to other mussels preferentially.
And it will keep the predators out of those larger beds. So that was yeah. Other questions in the room? You're going to let me ask one more? So I'm curious, you started this by talking about kind of the amnesia of the long-term shifts. You have a massive amount of data, but it's 20 years.
Mm-hmm.
So is there a chance that what we're seeing isn't actually represented for a long-term shift? Because if we're talking about many of the invertebrates are short-lived, but some of them are longer. And you talked about that 30-year mussel.
Mm-hmm.
So we're not quite at that lifetime stage yet.
Yeah, and there is a chance at the broader scale. I think the experimental nature of the study allows us to really get at the mechanisms very well, because we cleared everything to bare rock except for the control treatment. And so it is functionally primary succession. That is, this community patch is starting over. and we get the sense of what it is like over this long period of time. In reference to the control, which of course was not cleared, and yet we still saw changes in the control plots, not just the ones that we manipulated. I think that in the long run, these states are fairly persistent. And I want to go back and measure it after every stressor. This past year was a massive El Nino. And so I did actually see kelp declines in some places because of that. Now let's see, did they come back in 2025? So as the heat wave conditions continue, and they're only projected to increase, I do think that we're going to see more rapid change. And some of those changes are going to be so quick that the communities cannot bounce back from them. That's my short-term and long-term prognosis.
All right, one more from Scott. Good, because otherwise I'll just keep asking questions.
So did I interpret it right that we saw some diversity increases post marine heat wave?
Yes.
How would you fit that into like a intermediate disturbance hypothesis kind of framework of like, is that a net outcome here that we had a bit of stirring the pot and increase in diversity was a result of that?
That's a really good question. So in case you're not familiar with the intermediate disturbance hypothesis, it's essentially a curve that on one end of the spectrum, you have really low disturbance, and therefore you get species that are competitively dominant that can persist long-term. Those might be things like the mussels themselves that really thrive in this low disturbance. At the other end, you get so much disturbance that you only get the weedy species present. So you might think of like on the edge of a road, right? So you only have a handful of species that can really survive in those conditions. And somewhere in the middle is like the sweet spot of disturbance, such that you get some competitive, some weedy species. And I do think that there is some of that going on. I also think that it's a byproduct of how we can detect the species in our surveys. So I'm a lot better able to detect different species of invertebrates than I am all the possible species of, say, crustose coral and algae, which you would have to take samples of the crusts and send them in for sequencing. So when we sample some of these things, I do think we're not getting a perfect species detection situation. We are detecting much better the invertebrates than we are some of those crustose and cryptic species. So I think that's part of it as well.
Nice. All right, I think I've held you long enough. How about one more round of applause for Zach?
Thank you.
Thank you, everyone. Thank you for being here. Hopefully you come back next week and learn a little bit more about Alaska crab fisheries.
much in